Good afternoon. I'm Kennedy Aaron Aguon, and this afternoon I would like to share with you local knowledge of uh, fishes by the Lundayeh community. Right. This afternoon I'm going to present to you the local knowledge of freshwater fishes among Lundayeh ethnic in Pasia, Sabah, Malaysia. Introduction. Sabah, located at the northernmost of Borneo Island, is a country blessed with a high degree of flora, fauna, and indigenous ethnic diversity. Yanti reported that the number of freshwater fish species found in Sabah represented more than 30% of the entire Borneo Island. According to Marta, local knowledge on food plants and animals enables the utilization of these diverse natural resources. Local knowledge and freshwater fish use inherited from forebears are transcended orally and rarely written. Knowledge of food, plant, and animal species varies between indigenous native groups or communities and are highly dependent on the availability of plants and animals surrounding the domain. A literature review. Lundayeh is one of the 35 indigenous ethnicities in Sabah that live in the interior highland district of Sipitang, Enam, and Keningau, according to Lembaga Kebudayaan Negeri Sabah. Lundayeh categorized food plants and animals based on its function and role in daily sustenance. It is categorized based on Lundayeh food system, such as staple food, plant, and animal, alternative food source, vegetables, fruits, mushrooms, spices, and flavor enhancers. Or stated that Lundayeh had maintained the tradition of food through proper forest management and stewardship. Unrecorded local knowledge may be lost over time and even extinct. Therefore, written documentation is vital to prevent the loss of local knowledge. Arjiningram stated the mega diversity of non-kayu forest reserve and the massive local knowledge of the Lundayeh in Malinao, northern Kalimantan. This study aimed to identify and record the local knowledge of fish among Lundayeh of Long Pasia. Research methodology. The study was conducted in Long Pasia, Sipitang district, Sabah, Malaysia. It is a rural village located in the Borneo Highlands of Sabah at an elevation ranging between 1,000 to 1,600 meters and is populated by more than 90% Lundayeh ethnicity. The others are Murut, Kayan Kenya, Timorese, and Larantuka Adunara. And the main livelihood of Lupasia Lundayeh revolves around farming of fragrant Adan rice, rubber, palm oil smallholder, a gathering of jungle produce when in season, and hunting. The data collected by open-ended and semi-structural interview and explorative method following Martin. Primary informant and respondents were determined by purposive sampling method following Gide. The fish were identified following Moxie and Amba, and Inger and Sheen. Descriptive statistics were used to describe the number of freshwater fish species based on its usage. Findings and discussions. Matang and Pasia River characteristics. Generally, the rivers in study area were swift-flowing. The Matang is fast-flowing clear water with sandy bottom, gravel and rocky bottom, with forest canopy, while Pasia River is red, as Sia in Lundayeh language is red, turbid water from the tannin of tree leaf and bark, and the banks contain boulders and rocks. Riverbank vegetation consists of a primary rainforest. This observation is typical of a pristine Bornean high-altitude rainforest. The water temperature for Matang River ranges between 20 to 25 degrees Celsius, while Pasia River ranges from 18 to 23 degrees Celsius. These temperatures are lower as the study site has an elevation of 1,000 to 1,100 meters above sea level, and the rivers are covered in pristine tropical rainforest canopy. Fish species that were identified: 16 species of fish belonging to 4 families were identified. Analysis based on the number of individuals collected indicated that 60% were from family Gastromyzonidae, 30% from the family Cyprinidae, and 2% from the family Nemacillidae, and 1% from family Cirrosidae. Protomyzon, Gastromyzon, and 4 species were the dominant genera of freshwater fishes identified, as shown in Table 1. So under the family Cyprinidae, we have the tow, Uranices, Pontiucellae, and Paraprochycillus asterisk. While for family Gastromyzonidae, we have Gastromyzon bornensis, Monticola, Leptogaster, Fasciatus, and then we also have Glaniopsis hanitschi, and also Tinodata, Glaniopsis gossis. Then you have the Protomyzon pithedae, Bonensis, and Aphyloxylus, as well as Griswoldi. For family Cirrosidae, we have Galactothorax major, and family Nemacillidae, we have Nemacillius olivaceus. The species identified at the study site were low, probably attributed to altitude, since the study area was located more than 1,000 meters above sea level. Yanti reported similar observation, while Watson mentioned that freshwater species diversity of constituents seems to decline with altitude. Lower elevation at less than 600 meters would normally yield higher species richness but with smaller population size. Matang and Pasia rivers contain fishes that had evolved anatomically in order to adapt to fast-flowing, high-altitude water environment. As reported by Taylor, Gastromyzon, Protomyzon, and Demasialus species survive on macroalgae growing on rock surface, detritus, and insects. Presence of Gastromyzon as dominant species followed by Cyprinidae found in Mata and Pasia rivers suggests the excellent water quality with rich oxygenation and absence of human disturbance. As Long Pasia community also practice Tagal system introduced statewide by Sabah Fisheries Department. Further, this species might be useful as a bioindicator of river water quality in high elevation. Ismail described the dominance of the family Cyprinidae in lower riverine system all over Southeast Asian region. However, in Mata and Pasia River, it was mainly dominated by the Gastromyzontidae, as shown in Figure 1. Yanti observed that gastromyzontides would gradually replace cyprinids in freshwater succession at elevation above 600 meters. Inger and Chin mentioned that gastromyzontids are uniquely endemic to Borneo, such as gastromyzon and protomyzon species. For the rhodesienses found in Pasia, probably indicate that area could have ideal habitat conditions for the species to thrive. Those species are commonly found in swift, clear, and rocky bottom streams in pristine tropical rainforests at elevation exceeding 1,000 meters. Lundayeh community in Long Pasir adhere to Tagore system introduced by Sabah Fisheries Department statewide, which prohibits fishing during off-season and only allowed mass fishing once a year during open season. Small to medium-scale freshwater aquaculture cooperative might generate sustainable income in the community to culture these high-value table fish along the Matang and Pasia River banks as protein source. Taylor mentioned genus Pontius being traded as an ornamental fish. The freshwater fishes of Lumpasia had anatomically adapted to fast-flowing current with excellent water quality. Fishes living in those river conditions would generally exhibit less variety due to lower food source variability. Yanti explained that Gastromyzontidae successfully dominated uh, in higher elevation above 1,000 meters is due to its ability to graze on macroalgae growing on rocks. Therefore, it is of utmost importance to conserve Matang and Pasia River in Long Pasia pristine ecosphere for freshwater fish species. Conclusion: the pristine Matang and Pasia rivers and Long Pasia confluence plays crucial role in maintaining freshwater fishery stock in the study area. The fishes in Matang and Pasia rivers are dominated by the family Gastromyzontidae. This species had evolved to thrive in fast-flowing Bornean high-altitude rivers through several survival strategies. Cyprinid fish such as high-value table fish, tall species were found in the river study. The target system being practiced by Lumpasia Lundayeh community must be continued and further enhanced with the introduction of small to medium scale freshwater fish aquaculture to culture these exotic species. Acknowledgement. The authors are grateful to the entire Lundayeh community of Lumpasia for permission to conduct this preliminary research and their kind assistance. Authors would like to thank Mr. Ricky Jacob Gana for identification improvement and indigenous knowledge sharing. Authors wish to express our sincere appreciation to University of Malaysia Sabah for generously supporting this research through TB1914 PPIB Seed Money Research Scheme 2019. Thank you for your attention. Now we'll open the floor for question and answer. Thank you.